Welcome to Neurosalience, the OHBM podcast.
Today, my guest is Dr. Seong-Gi Kim. Seong-Gi is a kindred spirit. In the early days of fMRI, about everything related to fMRI contrast mechanism research, physiologic underpinnings, limits and methodology that I thought would be interesting to explore, Seong-Gi was ahead of me and produced results faster and better. Early on, he embraced the much more difficult but ultimately more penetrating work of fMRI on animal models. Seong-Gi is simply the best in the world at what he does, which has been for the past quarter century research on figuring out the interplay of pulse sequence weighting, neurophysiology, and fMRI contrast. Essentially, he's been leading the world, pushing the limits of our understanding of the biologic underpinnings of fMRI contrast towards answering systems neuroscience questions. Seong-Gi received his Ph.D. in physical chemistry from Washington University in 1988 and for investigating blood flow using NMR spectroscopy, and did a postdoctoral research fellowship at the University of Washington on determination of biomolecular structure by NMR. Dr. Kim moved to the Center for Magnetic Resonance Research at the University of Minnesota and joined the Human Functional MRI research team, which produced one of the first human fMRI papers in 1992. After spending a decade at the University of Minnesota and advancing to full professor, Dr. Kim moved to the University of Pittsburgh in 2002, where he was appointed as the Paul C. Lauterbur Chair and built a state-of-the-art neuroimaging center. In 2013, Dr. Kim moved back to Korea to direct the Center for Neuroscience Imaging Research and to be a faculty at the Sungkyunkwan University in Seoul. This is a pretty intense podcast that has a slightly different format than my typical podcasts. I hit on about 15 of the big questions in fMRI, from the pre and post undershoot, negative signal changes, new types of contrast, fMRI specificity, spatial and temporal resolution, and other things. Towards the end, we talk about his inspired work using optogenetics to provide insight into resting state fMRI, as well as how excitation and inhibition affects fMRI contrast. And now, here's Seong-Gi. All right, Seong-Gi, thanks for coming on.
Thanks for inviting me. Thank you.
Yeah, this will be fun. I just want to start by saying, just up front, that someone who has, you've been in the field for ever since the beginning, and in terms of the breadth of what you've done and the depth with which we looked, I can't think of anyone who's done as much as far as you're concerned. So yeah, and hopefully we'll get a chance to talk even about your recent papers, but this is a great opportunity to sort of pick your brain on a lot of things that you've touched on. You've touched on so many things. And I just want to, I usually start out by just asking, as far as how you got started, what made you interested in doing this? Was it just a matter of just being in the right place and just mentors that were significant or collaborators that were significant to you? I'm kind of curious.
Yeah, thank you, Peter. I think it's a great introduction. Thank you so much. Obviously, it's a lot of things, happen. It's not planned, those one. So when I went to the graduate school, it's Washington University at St. Louis. So I decided what to do about there. So I actually have something we talk about optics, and there's NMR lab. So I decided to do NMR research for my PhD program. So there was Joseph Ackerman, and many of you know him very well. He's the inventor of the surface core. So I went to his laboratory for my PhD work. So he gave me a lot of options what to do about those ones. So I actually chose to do perfusion measurement with NMR. So we inject the deuterated water into the animal. Then we use the deuterium NMR. We follow the washout of the deuterated water, and we calculate the perfusion value. So that was my PhD work. So I was, learn a lot about the perfusion and also NMR spectroscopy. I think this is one of the foundation of my lot of later perfusion research in my career also.
Okay.
And I think then I think I went to the 2 years postdoc at the Washington University of Washington at St. Louis. Seattle from structural NMR field. So nothing to do with the current my research those one. Then I went to the Minnesota. So I met a lot of people, and I think especially neuroscientists. I met Peter Strix, Georgiopoulos, Apostolos Georgiopoulos,
Yep.
and also I met Constantino Idokola. He's a vascular physiologist. So I worked with them, and I learned greatly from them. Is how to investigate the brain. I think this is a more likely foundation of my whole research line now.
Yeah, and right, being in Minnesota, coming in at just the right time, and so you were there. You arrived at just the very beginning of fMRI.
That's right. So that's I think I was at the Seattle. So it's Camille called me. Asked me about whether I'm interested to move to Minnesota. So I didn't know anything about MRI. So I went to the Minnesota, and Camille asked me to do, that's what I actually joined the University of Minnesota is October of the 1991. So it's that's what we started this, functional MRI at the time, those one. And yes. Camille assigned me to do the project. So that's it's a complete luck for me. It's Camille asked me, called me, and joined those one. I didn't know anything about MRI, and I went there, and he asked me to do the project. So what is that?
That worked out incredibly well. And
absolutely, completely my luck. And then I think Camille invited me to his CNIR. Is a great luck for me. I think it's
the right place. And actually, I feel I think talking to a lot of people. I mean, it's sort of talked to Bob Turner and Ravi and Ken Kwong, and even I feel the same way, that it's sort of like there's a lot of serendipity involved in a lot of this, and certainly you come in with some idea, and then you to the extent that you embrace it, it determines your career trajectory. And so, but then you quickly you became involved in doing fMRI in the early days, and you also, I mean, you were early on mostly known for having developed arterial spin labeling techniques. I mean, there certainly were some ideas floating around. There was a group at Stanford did some, Edelman had Epistar, but you came up with FAIR. And I know Ken Kwong had another one, and that was slightly different than FAIR. He didn't name it anything, but you came up with FAIR. And so maybe talk a little bit about that before we get into everything else. But because I think that was you did a lot of work with that, and it really helped push the field. And so how did you what was your process of coming up with it and pushing it?
So it's when I was at the University of Minnesota, so I got a professor position at the University of Minnesota. Camille gave me the position also. So I was just thinking about what to do as a faculty position also. So I needed to develop my independent research. So I was thinking about what to do about those ones. So then at that time, Bob Edelman is showing around his Epistar images, those one. Then I was already interested about the quantification. Anything about my background is all the perfusion or NMR field. I'm already interested in quantification. So I actually look at his data set, and I thought maybe we can actually do inversion recovery without it's a local, it's a global inversion versus a slice selective inversion that can come up with a perfusion contrast. So that's idea actually came watching his talks. When you actually watch his talk, that's what I got the idea of those one. Then I came back from the meeting to the Minnesota. I quickly implement. Then I actually implement very similar as what the Ken Kong's idea. I didn't know what the Ken Kong's work at the time. Okay, yeah. Apparently he actually presented the ISMRI meeting before those one. So I actually did a similar experiment what he actually tried to do it. Then it turned out did not work out very well. So I gave up quickly about the quantification aspect. So
Yes.
I decided to the fMRI study. So I had the sequence. That was actually even like Christmas time. So it's almost like a Christmas time. I got a foreigner. I work at the laboratory there. I actually asked my colleague going the magnet, and the first shot I actually got a very good data set.
Yeah.
So that's how we actually start. And then I quickly involved this, and I know how to do is a quantification. I got all the modeling, those one. So I do then I come up with what is a acronym, because it's very important. When I was a postdoc and I developed a new technology, but I did not put any acronym, it did not turn out very well, even though there was very good idea. So I was thinking about how I can make an acronym those one. So I come up with the idea. Yeah, that's what it's. So that is turned out to be a great success. Yeah.
Yeah, that was smart. That was a good idea to put an acronym on it, because it actually, yeah, I mean, certainly now there's an entire slew of acronyms with various tagging schemes and acquisition schemes. But I mean, the principle of FAIR, was very elegant in the idea of just doing a global inversion versus a single slice, and getting or the local inversion to get all the and then subtracting. That concept is still used today, and yeah, I think it is important to note that you spent a lot of work, a lot of time working on doing actual quantification of perfusion with that. And it's funny because I just have, without going into the details of the quantification of it, I just remember there was a lot of discussion as to whether you could quantify and how many assumptions you had to make, and you had more of a T1 based sort of analytic quantification approach. And I think I remember, actually, I still remember like Eric Wong and yourself having an argument at one of the ISMRM meetings, because he had more of a kinetic model, and there was, I mean, obviously there's a convergence, but still it was a really, I remember it was an intense time as far as that's concerned.
Yeah, so it's I was very reluctant to quantify the value, because we know there's a lots of problem about the quantification, because all that depending on modeling approach, whether like water is fully diffusible or not. This is a big issue about that assumption coming down those one. So we assume the water is freely diffusible. Then that's what is a T1 model coming down. So that's a lot of discussion those one. Still, there was issues already there. So when you quantify the value, this whether we have proper assumption or not, this is all issue. So I almost convinced. Then I said, okay, let's try to do the relative change. So you see, the most of my actual research about the FAIR is to determine relative blood flow change during the stimulation.
Yes.
That's how I, it's a guide my research in there. Then I try to do also oxygen consumption changes, measuring is a perfusion and the bolts, and then is hypochemic calibration, something like that.
Yeah, right around 2000, I remember Rick Hogue also working on this, and you were working on it, to sort of having a CO2 calibration. We have a global flow and without activation, and then you get the ratio of flow to bold, and then you can extract. I mean, this is what you have done so well, is use this deep knowledge of pulse sequences and physiology to sort of with this creativity in terms of looking at the ratio of flow and bold to infer metabolic rate. And attempt to and try to get metabolic rate information. It's a hard experiment, but it was also useful. Yeah.
So I can just give you a little bit background those ones. So I think once we actually have, it's a FAIR technique, I immediately want to do the CNIR optimism. So I combined the FAIR and the bold together. So we actually published a paper about the CNIR estimation. Based on it's like a M factor, whatever we estimate about based on the model those one. Then it's Minnesota. There was 40 was down at the time. Okay, some time point was 40 was down. So I went to Denmark to do it's a hyperchemic experiment.
Okay.
So it's Denmark. There was all of persons labs. So they do a lot of ways. It's a hyperchemic study. So I went to his laboratory. Then it's trying to do the hyperchemic study, and then I found that there was a winter time. I went there. I found the paper coming from MGH. There was a first hyperchemic paper. Those. So that's how evolved that field.
Okay. And it's amazing how these things come up in parallel like this. At a certain time, they sort of come along and right. I mean, I think, and yeah, that's right. You were, so I noticed that paper, right? You were, Albert Getty, maybe I think was on it, but yeah, Olaf Paulson.
There was Olaf Paulson was there. So I have a couple of papers with Olaf Paulson. There was
Okay.
not Albert Getty, so Paulson was there.
Okay. Great. Yeah. And so back then, so just before we get into all the other points that I wanted to bring up, did you have a sense of how big fMRI would become?
I mean, at the time, I never thought about that big those one.
Right.
It's we have all the scanner we are doing is a very limited, very few center can do that kind of study. Those one, and not many cognitive neuroscientists are engaged those one. I mean, I work with neuroscientists, system neuroscientists. They do the same thing, and we actually work with collaborating people. Those one, there are some sense fMRI. They're kind of they do the fMRI study, but kind of reluctant. It's not likely, really believe it or so much. Those one, so there are already some concern. Those one, so I never thought about that big. But this is just amazing, how big it's right now, and I'm director of the center, or this,
Yeah. No, I think you're right. I think that at the time you're sort of like just thinking really hard about, the current, climate of the certain questions that are there, and without sort of sitting back and thinking about, the overall thing. But yeah, and it was so intense, and it's still intense now in a different way. It keeps on changing. So, all right. So I just want to, all of these topics, I have like 15 things related to issues and controversies and questions about fMRI. And, it's truly impressive. You've sort of, all your work has sort of touched on all of these. And what's even more impressive is that, and you always were comfortable with this, and I never did, and a lot of people haven't, is jumping into the animal models. And you've done that so well. And there's so much more information you can get from the animal models as far as that's concerned. But let's just start with maybe somewhat chronologically, not really, but the pre-undershoot. So that's a big thing in fMRI. There was controversy. They saw it, they didn't see it. You were the only person that I saw was able to actually modulate it by controlling blood pressure, which was really impressive. Do you want to maybe talk about that? What is the pre-undershoot, and why do we sometimes see it and sometimes not see it?
So yeah, I think so. It's a tip. Is a pre-undershoot. Is a tip. Is a very hard issue. I mean, early late 1990s, early 2000. That was a very hard issue. Those one, and the American Green Board publishes optical imaging paper, and we already thought is early dip is the answer for localization.
Yeah.
Right?
Yeah.
So I actually started the fMRI study. So I have two line of research I did at the time. So one is the use of rat models. Alfonso Silva, he was working on those project, and we never see anything about tips. Okay,
yep.
No. So in the Minnesota was the center of the origin of the tip in the sense. So it's Ravi working on those one, Sharpen who was working on the tip those one. So I was skeptical about the tip at the time because I could not believe because I didn't see those one.
Yeah.
Then is one hand we got Seong-Gi Kim. So he did is a cat study, and then Daeshik and Tim Duang, they are working on this cat study, and we actually see the tip. Okay, I mean, so it's a tip, and we map it out. Looks like a column. Looks very good.
Yeah.
And then what is it? Then we have a lot of controversy about this, whether this is reliable or not. We know there's Nature Neuroscience. There's the letters all there. So for me, it's imperative to actually reproduce the data set. Well, I mean, it's very important to reproduce data set. So we're working very hard to trying to figure out what to do. So there's a lot of variation. Sometimes you do see the tip, sometimes you don't see the tip.
Yeah.
So one time I actually watched the experiment, and one animal, we actually see the tip, sometimes we don't see the tip. So I watch what's happening those one. They are related to the blood pressures.
Yeah.
whatever reasons. Okay. So I thought it's okay, we don't know what is the reason. It's a blood pressure changes that cause the problem.
Yeah.
So I actually decided to use to modulate the blood pressure. So called the sodium peroxide. So we infuse the sodium peroxide. Then this is is a dilated vessel.
Yes.
Then when you stimulate, there's no further dilation. But consequently, it's like low blood pressure.
Yes.
So you can actually modulate, increase dip. This is a complete dip, essentially. There is no even positive change also.
Yeah.
So you can make a complete dip. So there is no blood flow changes, no blood volume change. There's no only oxygen consumption changes. So there is a dip in this, huge dip in the sense. Or
Yes.
it's just negative change.
Yeah.
So dip is originate from oxygen consumption change. There's no doubt about this. So there's some people talk about the maybe venous blood volume changes. That's not true. Venous blood volume changes actually quite slow. Those one that's not happening. Those one. So depending on CMR2 changes and the blood flow changes, is a normal lot of condition. Blood flow response very quick, very fast. Then you don't see any dip.
Yes. Yeah.
But in some case, it's a disease condition or like an anesthetized condition, animal case, those one, then blood flow response are quite slow sometimes. Then you do see that.
Okay.
But you could modulate. So I think there was an interaction between, CMR2 changes and the blood flow change.
Yeah. And who knows, maybe in humans, it may have been the subjects had maybe a little bit lower blood pressure, or who knows? I mean, maybe, that's never, I haven't seen the literature of that being explored in humans. So, I mean, it can certainly be done. It would be cool to see, to try to reproduce that in some level.
Well, I think a certain patient groups, you might have it. I mean, you don't want to reduce the blood pressure because that's what you wanted, okay?
It's so low. I mean, you're right. You really modulated the blood pressure in these.
We actually modulate. We almost make it's a 15 millimeter mercury. That was our target value. So normally the blood pressure 100 millimeter mercury. So we reduce a 50 millimeter mercury.
Wow. Okay. All right. And yeah, it was, and that actually shows that there's no like rate pulling oxygen from the blood there. So that was cool. So maybe before I get into negative signal changes, might as well naturally go to the post undershoot then. I mean, you've also worked on that. And there's also some work, suggesting, as you mentioned initially, that the initial dip is maybe more localized because it's without any penumbra of spilling over oxygenated blood. You have this oxygenation change that's localized. You also did some work on the post undershoot, understanding it. And you have a paper in 2007 in Neuroimage that suggests that, it's potentially more localized as well. And also suggesting that the origin is more I'll let you describe it. Okay. What you think the origin is. Yeah.
Okay. So we in the cat it's an optical cat MR study. So we have to use a cat visual cortical area to look at the property of the ball signal, and we do see the huge post-stimulus undershoot. Yeah. Okay, I mean this is something we actually see those one. Then we actually look at the localization. This, post-stimulus undershoot showed improved spatial specificity relative to middle of cortex. So it's actually pretty good also. And the source question is what is the source?
Yes. Yeah.
Source is one, obviously, there's a prolonged blood volume change. That is a possible.
Yeah.
Okay, if there's a prolonged blood volume change, that is also possible. The other one is oxygen consumption change. Somehow oxygen consumption change during the stimulation period, but blood vessel area, they are prolonged effect. So it's maybe like combination of the oxygen consumption changes happening during the stimulation period, and then is a prolonged blood volume change. Because blood volume change appear to be specific.
Okay. So you think it's so right? There was this.
It's complicated.
It's complicated.
Yeah, it's complicated. Post-stimulus undershoot is a complicated. I don't really, so like a lot of 3 different possibility, blood flow actually decrease during stimulation period, prolong the venous blood volume increase,
yep,
and oxygen consumption changes. So 3 different parameters there, and it's a complicated. I think depending on what the situation, there can be different players.
Yeah. Okay. And that's a good, right. That's sort of my conclusion as well, because it seems like, there's, I know Peter Wenzel, argues, metabolic perseveration, but you're right, blood volume is definitely also continues on a little bit longer. Some people even claim that there's a flow dip as well, but like you said,
That's right.
It might be all of those, and it might vary over space. It might vary with resolution. Who knows, right?
I mean, so I think it also depending on what the stimulus you do. I mean, you do the study there, or like you do the very long stimulus or short stimulus, and the strength of the stimulus. I think there's a lot of things. It's very hard to make one single conclusion.
Yeah. Yeah, I think there's still room to work there. I mean, I remember I was able to modulate the post-undershoot pretty strongly by changing, back, in the late 90s, sort of with a red stimulus versus a black and white stimulus. The red stimulus had a larger post-undershoot. It was still sort of curious why that would be, but it seems like there's potentially work there, and also even might be relevant to your work on inhibitory activity as well. Who knows? But okay, well, that was your touching on that, and I think that's one of these issues that's still somewhat it's more complicated than we're able to tease apart yet. So now the negative signal changes. You had a really nice paper in Journal of Cerebral Blood Flow and Metabolism in 2002 talking about negative changes, and it was one of the first ones that sort of addressed it, and everyone was wondering, what are these negative changes? You actually came out with an interesting hypothesis that was pretty well argued, that it's sort of a potentially a steal effect to some degree, that there was some pooling of blood from other areas that might be a steal effect. But at the same time, I think that there's recent papers from you as well that might suggest there's, it's a real, decrease in neural activity. So what do you think it is? What did you think it is? Has that changed, or is that, is there, once again, it could be complicated as well?
I think there's a post condition can exist. In the most of cases, there, inhibition cause decrease of blood signal. So that's what is the most of cases. But in some cases, for example, like a disease condition or anesthetic condition, there is a possibility there is a stealing effect, because that is a common. I did not make the term of a stealing effect, because I found that the terminology from the physiology literature. That's what it's in the patient study. They actually show the stealing effect. Yeah. So I think it's the most of case we do. I think most of us OHBM people, they do the normal volunteer. Then I think it's a negative change assumed to be the inhibition. Yeah. Or like what is in the patient study, somebody do in the patient side, then this is a little bit different story.
Yeah, where the neurovascular coupling is somewhat altered, or right.
Neurovascular coupling is altered, their vascular response not happening very easily, something modulated in the sense. Right.
Yeah. And it brings out the point that you have to be very careful with patient populations as far as interpreting the signal. But it is interesting that, right, we see negative signal changes all the time in healthy volunteers. And there's something real going on there in terms of people don't as much look at that, but they obviously have been doing a lot. Yeah.
But another point, actually, probably want to point out those one. Negative change can happen in the large vessel area. Okay, that is one of the reasons is when we actually do the experiment, it's brain actually a little bit is expand. Okay, then sensor volume is actually decrease.
Yep.
So you got edges those one. Edges actually can decrease is a board signal depending on is like a partial volume change.
Yes.
Of the CSF and the brain those.
Yeah.
So that area is have to be very careful about how you interpret data.
Yeah, that's a really good point, and especially that very high resolution too. You get this have this more pronounced effect, and yeah.
So when we do the animal study, we actually measure these things there, and we estimate about 5 micrometers or something like that. Very small is swelling in there, and then there is a big changes in the MR signal.
Well, that's interesting. So the brain actually does expand with maybe the increased blood volume, I guess, a little bit pushing things around a little bit, or yeah.
So what we do though, we actually do the spin locking experiment. So it's a T1 low experiment there.
Yes.
So we actually suppress it's a brain signal. We only keep the CSF signal. Then we do the activation style. Then you can actually see it, how it changes.
Oh, really? Okay. Oh, that's interesting. So the T1 row is, I was going to get into that, but yeah, so T1 row is something that, without going into the physics of it, it actually does suppresses the tissue.
Well, we got a T1 row is of the tissues much shorter than is a CSF, just like just like T2. These are very similar to T2.
Yes.
So we just use the T1 row as the tool set to suppress a gray matter signal and then maintain its CSF.
Okay. And you noticed that so when you did that, you actually did notice that the CSF sort of repositioned itself in some sense.
Yeah. I think we published this one is neuroimages. I think I don't know, it's 2010 or 11, something like that. And so there was a CSF volume changes.
Yeah. Okay. All right. I'll have to look at it. We should start having show notes for this as well. I realize that there's so many cool papers that we talk about, and you just have a whole list of ones that are great. Okay. All right. So that's also a potential source of that negative signal change. So yeah, something to keep in mind as well. So, all right. So then also you wrote, and this is something that people are still debating now. And I actually just saw this paper. I didn't see it. It was a very recent paper, really beautiful review article in the Philosophical Transactions in the Royal Society B, where you kind of go over the spatial specificity and you also talk a little bit about the temporal resolution too, but just review the literature really well, including your own work. So what's your thought now in terms of the upper limits of functional imaging? It seems like you have a number there on the order of blood is regulated at about 0.5 millimeters. And of course, different sequences have different point spread functions depending on what hemodynamics they're looking at. So what is the upper limit in terms of spatial resolution, at least?
Well, I think I just want to talk about the animal, because I think I don't know this. We can extrapolate directly to the human very easily on the animal study. Yeah. When we look at the capillary change, the assumption is we already look at the microvessel changes. Then it's a blood flow and possibly also blood volume changes. We are looking for small vesicle changes, vessel changes also. Those areas is very easily we can achieve 500 micron as a specificity. Even like a blood volume, we are getting 100 microns.
Okay.
So blood volume is a very specific in mouse. Okay, mouse we are actually getting about 100 micron full width half maximum spread relative to neural active site.
Okay.
And in the blood flow we could not measure that kind of blood flow because sensitivity is not there. Yeah. So it's very hard to measure those one. But when we look at the column study or layer study, we can actually see about the 500 micron, localization.
Okay, so it is interesting that let's say you're doing spin echo with a short readout window. I think you have a flash sequence, so you don't have to worry about the wide readout window with echoplanar. Pure spin echo, and assuming that you're at a high enough field strength that you don't have any intravascular blood effects, how does spin echo compare with, how does capillary bold compare with venous blood volume effects? I mean, not venous, but vaso or something like that.
Usually very similar. I mean, that's what I might guess. In this instance, when you do the really true spin echo measurement, should be very similar. But I never tried to the true, like a spin echo measurement. So already when we do the EPI measurement, then there was a window we have there waiting function also. So in that case, is a spin echo is not as good as the blood volume response.
Yeah.
But we actually have a true one, then I think the spin echo should be very good.
Yeah. Yeah. And it sort of speaks to, I think bold is probably you're more sensitive, even with the spin echo, maybe than vaso, maybe similar, but it's interesting. It sort of speaks to, and I always wondered, I'm still wondering about this, is that why is, blood volume, it seems like capillaries, there might be, capillary recruitment or something like that that change blood volume, but it seems like blood volume is a little bit upstream, and that seems like it should be slightly less localized where you have sphincters changing. And it always seems that it's not as much upstream as I thought. And so, and you had a paper that sort of separated out arterial blood volume versus, capillary blood volume. But I don't know, what are your thoughts on, it seems that it works.
I think you are right in the sense, because I think we need to look at the weighting function. Okay, how much weighting coming from the arterioles or like penetrating arterioles in the sense, and how much you coming from the capillary, how much you got the venous in the time domain wise. Because I think when we stimulate, so I just look at what explained by experiment. Yes, when you stimulate, we do see penetrating vessel dilate first. So you can see there is no localization across the layer. So even if you actually stimulate the layer 4 input coming in there, you don't see highest response layer 4. So all the layer almost like a similar. First, probably like when you stop. Yeah. Then a little bit later on, then it's a microvessel start to dilate.
Yes.
I don't know the reason basis there. There can be active dilation by somehow it's a vasodilator, or the passive dilation by a large vessel, there's pressure in there, and then dilate also. Then you can see start to it's a small vessel, or possibly the capillary is a dilate over the time. Then you wait longer, you are going to have a better spatial localization. First, if you look at the first 10 seconds, it's not very well localized.
Yeah.
You have to wait about the 10 seconds later. Then you got a weighting that changes.
Yeah.
Okay, weighting is more weighting coming to the small vessel.
Interesting.
Then apparently large vessel is a initial dilate one is not dilate anymore. So it's essentially when we look at the dynamic property there, then it's a large vessel initially dilate, return to almost like a baseline condition. Then it's a microvessel keep dilating.
Yes. Yeah.
Okay.
Okay.
So this is somehow is a time window is very important.
Yes.
So when you look at the optical imaging paper never say blood volume is a good choice. They always complain about the blood volume is bad. You have to use a dip.
Interesting.
Okay. Reason is optical imaging study. They already looking for a very short time window.
Yeah.
When you look at a very short time window, then you have a large vessel contribution there. Yep. So you don't have any special localization.
Yeah, that's interesting.
Okay, so it's an MR, we cannot scan that fast. Or like we have everything, it's a vessel, we also waiting there. So we tend to have a very long stimulation during the, it's a blood volume study. We do 20 seconds, maybe even 40 seconds stimulation period. So when you do the waiting, you are waiting in a longer time period, more than early time period.
Yes.
So it's a blood volume looks much better than what you expected.
Yeah. Yeah. Okay. Okay. I see what you're saying.
Understandable in the sense?
Yeah. That's interesting. And that's very interesting. That's a good insight as far as the blood volume effect. But then, and maybe we'll go right into the temporal limits. When you talk about bold, there's some suggestion that they're early bold because that's downstream already. That might be more localized too. So.
That's right. So bold is a different story. Bold is always early part is much more localized than later on. We all know bold starting from like a capillary to the draining vein side there. So whenever you capture early response, that's where it occur in the capillary area. Then that is the most localized. Yes. So it's a different from it's like a blood volume mechanism. Blood volume mechanism almost like to me like a later time point is better off, rather than very early time.
Yeah. Okay. All right. Well, cool. So that sort of covers as far as you wrote some papers on the upper limits of temporal resolution with Ravi, and I think you've sort of spoken to that in some sense. This, the pre-undershoot happens within a half a second, but then of course, if you have infinite signal to noise, you can actually pull out any temporal resolution you want, it seems, if you've modeled the signal well enough. But the spatial resolution is really the main work that you've done, and also it does speak to the temporal resolution in that regard too. If you're more selective with your capillary effects, you can actually push the temporal resolution very high, and obviously it's more sensitivity based as well. So let's get into I thought was the really cool parts of what your recent work has been. There's a big question as to, with going back to like just bold contrast or whatever, what influences it? And so pulling out, and I think your papers are some of the only ones of pulling out inhibition versus excitation. And you have a lot of, as I'm reading your papers, there's a lot of pieces of information about the signatures of excitation versus inhibition. And you've used optogenetics to sort of pull these out. Do you want to speak to that a little bit? What are your main findings as far as that's concerned? Because it's really cool.
Yeah, I think it's we are all talk about the excitatory cell activity. So I think this is understandable also. But inhibitory cells also is there. So about 20% is total cell population. So it's not a negligible, important contributor to the neural control also. Question is what is the physical response? Whether they are significant to our measurement or not, those ones. So obviously inhibitory neuron also energy consuming process that also produce vasodilators or vasoconstrictors. So is inhibitory neuron play role? Question is how much? What is going to happen those? So there was some of the literature. Actually my lab actually did also experiment to the optical imaging study. So you can actually stimulate the inhibitory neuron optogenetically, and you will see how the response coming down there. Then when you do optogenetic stimulation in the inhibitory neuron, you can actually see the increased Ball signal. Okay, because there is a blood flow increase.
Yes.
Okay, there is no doubt those.
Yes.
Now, so in the real case, so you have when you do the sensory stimulation, whatever we have it also, and then we are obviously going to have excited neural activity and the inhibitory neural activity come together. Right. So we cannot differentiate. So like, but when you optogenetic, you can kind of differentiate. When you stimulate excitatory neuron only, when you stimulate inhibitory neuron only, but this inhibitory neuron also interact with excitatory neuron. So it's still a complex issue in the sense.
Yes.
So
yes.
I can tell you that when you just stimulate inhibitory neuron by itself, you increase the blood flow. Okay. So when you in the cortical area, when you stimulate inhibitory neuron, then initially there is increased blood flow. Then inhibitory neuron interact with excitatory neuron, then decrease the blood flow. So you have a biphasic response. So initially positive response, then there is a negative response by the inhibition.
Okay.
So essentially that's what is there. But in the normal case, we don't know that is happen or not, because this is a case we actually hitting to the inhibitory neuron very hard.
Yeah. Yes.
Okay, then you maximize inhibitory neural activity. But normal, what we are doing now is the actual stimulation study. Then there is only part of the inhibitory neuron involved, and we don't know what contribution to the fMRI response. So when you characterize, our inhibitory neuron based hemodynamic response, excitatory neuron hemodynamic response, inhibitory neuron hemodynamic response appear to be faster than excitatory neuron driven it's a hemodynamic response. The question is, I don't know why, is a faster, they're probably a faster, somehow it's a vasomodulator or something like that. There is a nitric oxide released by the inhibitory neuron, then they hit it, it's a vessel right away, they dilate quickly those.
Yeah, that's interesting. It's right. So there might be a different mechanism, like, using nitric oxide versus maybe a neurogenic mechanism with excitatory, but it could be interesting then. So then
definitely there is different vasoregulator. That's the definite, that's a different person.
Okay. That's interesting in itself. And have you tried, it seems like it would be interesting then to maybe look at the flow to bold, flow to bold ratio to maybe pull out. It's hard to spatially differentiate because there's so much mix as far as looking at trying to quantify metabolic rate or looking at flow to bold ratio with, to map out areas maybe where there's a little bit more inhibition or less inhibition with that.
Okay, we didn't do that, but we actually, in the olfactory bulb study, you have some, I mean, so I think a certain brain area, it's a striatum, or like it's olfactory bulb, some granular layer, there is only inhibitory neuron.
Okay.
So if we want to investigate those one, we can actually see it. It's just inhibiting neuron area. So there is a obvious increase blood flow. Also, when you activate those inhibited blood flow, there's also oxygen consumption increase. But in the cortical area, they're all mixed together. There's 80% excited neuron, 20% inhibitory neuron. They all interact with each other. So it's very hard to tease out.
Yeah.
So if you want to tease out, then we have to use a different, area. There's a subcortical area or olfactory bulb. Then people already complain about this not part of the cortical area. So
yes. Yeah. And so there's right. Okay. So it's still a hard problem, but at least you're getting some hooks into it.
Yeah, how to do it. And then another thing is, I think it's interesting point you have to look at those. And the inhibitory neuron, there's a lot of subtypes. So the called like PB neuron, some of the statin neuron, MPY, nitric oxide. There's a subunit those one, and this subunit behave differently to the hemodynamic response. PB neuron supposed to be the most popular, it's most common PB neuron, parvalbumin neuron. Does not produce any vasodilators. So it's when you PB neuron probably does not change any hemodynamic response.
Interesting.
So it's quite complicated. Interneuron is quite complicated.
Yeah, and the temporal dynamics are so rapid that it's hard to pull out anything, using the transients of bold or things like that. I know that, Camille Uludağ had an interesting paper sort of trying to suggest that certain transients might be action of inhibition or whatever, but it's so hard to do that as well. I mean, there's, excitation, and there's maybe a wave of inhibition, and it all happens, and it's
all happening. And then this measurement is very limited. You have only one measurement, so you need to have a deconvolute those measurement with a different information those one, and may not be right.
Yeah. Okay, so all right. Well, that's the state of it, though. But I your papers, though, I have not seen people try to address this, and you do make some real interesting, as you were mentioning, the observations about the relationship between the flow and metabolism for both. So that's a start, and that's cool. So okay, so why don't we jump to what's your conclusion as far as if you have a paralyzed animal versus anesthetized, and then anesthetized with the various types of anesthetic, what gives people, it's hard to say most reliable because it all depends on their question, but what would you say is your summary of your work with various anesthetics and comparing to awake? I mean, because you work with that so much with your animal models that you probably thought a lot about that.
When I was a state, whenever we actually submit the NIH grant proposal, there is always a complaint about the anesthesia. You use an anesthetized animal and how you interpret your data to the human. So there was, always a question those one. So there was some anesthesia better than others. I mean, in terms of some is a very close awake condition. People use called dexamethasone a lot for mimic the awake condition. So it's like that's one. And then some of anesthesia is GABA agonist, like isoflurane GABA agonist. Then there's a suppressed neural activity as well as hemodynamic response. So that is not easy to interpret data set. And then we use a lot of ketamine xylazine. Ketamine is an NMDA antagonist, those one. This one is actually a little bit disinhibition. So we like this one because we actually see a lot of activity in the cortical area. So easy to detect the fMRI signal. So direct translation from animal data to awake or human condition have to be careful. All right, but we have to take an essence of the finding. Whether what is the true finding coming up? Okay, capillary response is specific, or how specific, when is happening. Those one is a change in the different anesthesia will change in different time domain, but general trend, general idea is going to be very similar. So how much will be localized? Maybe awake conditions maybe a little bit less localized, but blood volume better choice, then you got blood volume is a better choice. So I think you still need to have a recalibrate somehow in the human.
Yes.
But I think you got a general concept or idea can be translated. That's how I see those.
Okay. Okay.
I don't know if it's clear to you or something like that.
Yeah. No, it is clear. Right. Each one does a certain thing, and that it might make a difference in some cases, but it may not make a difference.
It might make a difference. There's obviously, it's awake condition is obviously the best way to do experiment, but problem is not easy to do awake condition.
Yeah.
Because a lot of time fMRI study, we need to have a very high signal to noise. We need a very high spatial resolution because brain is very small. So if you want to try to match the human resolution, human brain resolution, then we need to have a much smaller voxel size.
Yes.
So sensitivity very low.
Yeah.
And then we need to do a lot of averaging. So without averaging, it's very hard to investigate anything. So then the awake condition animals moving, so it's very hard to. You can train them.
But
we did those. We actually published one paper, awake versus an inside animal.
Yes.
After one study, I say, okay, that's it. And I don't want to do anymore. It take a long time, and I did not find anything is actually much different. Finding is not a whole lot different what I'm interested in.
Yeah, yeah. Well, that's telling in itself, and it's helpful, right? Right, and it's like you said, it's a really hard experiment to make a really fair comparison. Yeah.
Well, if really people want to do it, or like their life is basically, I want to have a awake is behaving animal. So awake is not important. We need to have a behaving animal. So if you want to do the real neuroscience type of question, you need to have a mice inside the magnet. They do the awake and the behaving. How you can do the behaving animal inside the magnet? It's very hard to do it, and you can do a certain experiment. But most people don't want, most neuroscientists don't want to do that kind of stuff.
Yes. Yeah. Yeah. No, that's a good point. So that's at least comforting in some sense with the people who are doing animal models, that there's not, I mean, there's, you have to really understand the anesthetic and what the effects are. But for the most part, for instance, using ketamine is a reasonable thing to do as far as that's concerned. So, all right, well, let's get on to other things. As we go down the list, we're getting along. So not to bring up any too much controversy, but you actually, I mean, I had Denis Lebihan on the podcast, and he's obviously done a lot of great work, and he's a very big proponent. He believes strongly in the idea that diffusion coefficient goes down slightly with activation because intracellular intracellular the cells expand, and you have near the membrane you have more low diffusion spins. You had one paper, we've tried to look for it, we couldn't see it that well, and you had one paper that suggested it was more vascular. And what are your thoughts on that?
I know this topic very well, and when Lebihan published a paper, after that we actually saw some studies of ADC measurement. So I think there is a couple of issues. One is obviously vascular contribution, one factor, but you can remove it. If you use very high p value, you can remove it. You still have ADC change, even after you remove.
Okay.
It's a vascular component. Question is, what is that?
Yes.
Is that truly coming from cellular signal or something else? That's, I think, the main question is to me.
Yes.
So we did not publish, but we actually did the experiment. Then I decided to just close that kind of project in my lab. Then we did not actually, we stopped the project. Then we end up the project also. But I can tell you what the study we did in there. So we did the ADC measurement in this animal. Then we inject the contrast agent. We can get rid of the intravascular signal. So there was no intravascular signal. So we purely looking for like extravascular in the tissue signal.
Yes.
Then what we did is a hypercapnia study. And as well as like a stimulation study. When we do see the ADC change, hypercapnia, as well as functional study.
Yes.
Or like
Yes.
this is the same thing as a Carla-Miller soil.
Right. Exactly.
Or like, so my question is, why happening?
Yeah.
So so we actually did we applied it's a gradient different direction. It's a different direction in the basically is a hypercapnia study. We actually applied a different direction. We want to see whether directional component. We do see the directional component.
Huh.
Reason is what we are thinking is a vessel is dilate. Okay, then intravascular is based extravascular pool. They are distorted, shapes changes.
Huh.
The vessel is dilate. Then there was a fixed space. There was one. Then extravascular is a space is going to be a little bit distorted.
Yeah.
Boundaries changes.
But that extravascular space, it still seems like that space is still high enough diffusion coefficient that would be gone in the high B factor, like if it's CSF or something. Or,
so intravascular is gone, but
Yeah.
extravascular signal is still there.
okay, so it's not CSF or anything. It's
Still
something.
there,
It's tissue.
but it's, when you distort it there, then it's ADC changes, diffusion coefficient changes because the boundaries changes.
Okay. Okay.
You see that? So ADC is a change during the stimulation, but most likely due to morphological change by the blood vessel, the blood wall changes.
Yeah. Well, that's
see my
interesting.
point?
Okay, that's a reasonable hypothesis. Although, so it seems like you can address that by doing change, like you said, changing the direction of the gradients, doing isotropic.
Yeah, we actually changed the direction, and we actually saw is the directional component. Some direction is much more than others. What that means is that direction is with morphological changes.
That's
okay.
That's
So
interesting.
we actually did that experiment. We almost like dropped this, paper. Then sometime point I say, okay, diffusion, we are not interested anymore. We want to just move on to the next project. So we didn't publish the paper, but that's what our finding. Then after that, I think we never touched anything about ADC.
Okay. And that's interesting. So it's not the intravascular signal, it's not the blood, it's just morphologic changes of the tissue being compressed.
Extravascular, extracellular space.
Yes.
Okay,
yes.
something like that. Okay, I don't know. That is my idea. And another thing is a very low sensitivity. Sensitivity is so poor. Is not as good as anything else. What I'm going to use in the sense, much worse than is a perfusion. Much worse than is a blood volume study, and I don't want to use diffusion study. Even they measure, like intracellular ADC changes.
Okay. All right. Well, that was new information. I think that, like I said, I think a lot of people don't necessarily. I mean, there's so much literature out there, and it's hard for anyone to follow all of it. And that was useful to know.
Well, there's something to think about also, right? Something to think about, how is ADC change during the hypercapnia?
Yeah. Unless there's a neural component to hypercapnia.
Well, people are talking about neural component, but there was not really.
Yeah. That's interesting. Yeah. No, I see what you're saying. I mean, certainly hypercapnia is a beautiful way of testing that, and No, that's that's interesting. So, okay, well, let's keep the controversy going. So how about other types of contrast? Do you see, something that I care about a little bit is somewhat, maybe I've dropped it recently just because I felt that it wasn't possible, or it might not be possible yet, but it's still there, I think, is the idea of neural current imaging. Have you ever in your experiments seen anything that might be suggestive that your animal studies or whatever of neural current effects or effects that are non-bold? Let's just say non-bold. Yeah.
Well, you tried a lot about the direct current measurement also, and I did not try by myself because I figured out sensitivity is very low.
Yeah.
It's not going to be practical. Or like even if I can detect, it's maybe there. So it's hard to use for my research. My point is I don't want to chasing around those. So I did not chasing around direct it's a measurement, but I actually did like a non-bold measurement with a spin log measurement. So I mentioned about spin log dose one. So if you remove all the vascular component, you can actually do the spin log study, And then we do see the actually it's a cellular component.
Okay.
And we are trying to see what exactly is not related to vascular one, how we can get a spin log or like T1 low measurement. We do see those one. So we think it's related to it's a chemical exchange.
Oh, really?
Glucose, hydrogen, and it's a water proton. They are exchanged. We know there's a glucose is there's exchange between chemical exchange between glucose hydroxyl group and the water proton.
Yeah.
Right.
Yeah.
And then when you have concentration changes or pH changes, this exchange rate is going to change.
Okay. Okay. That's really interesting.
Yeah. So we measure. So we actually probably published 2013 or something like a neuroimaging about the non-board effect or so. And it's there. And then we actually trying to utilize this idea to do, like more like routine experiment. So question is, can you actually use a glucose sensitive fMRI study to detect, metabolism area? And that's kind of idea we actually there. So we use a like spin-off study tuned to the glucose, for example, glucose frequency, those one. And we did a lot of follow-up study, even at the 15.2 Tesla.
Yeah.
We could not detect. It's a reliable.
Yeah.
I mean, when we do that experiment, we do crazy averaging in the sense. So the conceptual is working very well, but percentage is very low.
Okay. Okay.
So it's not easy to utilize this idea for our measurement.
Okay. Okay.
There was it's like, for example, it's a Neurospin actually published one of paper about glucose CEST fMRI.
Okay.
I don't know if you remember those one. It's kind of similar type of idea.
Yeah. 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 Okay. So you're looking at the exchange, and it's actually affecting the signal enough that, obviously it's extremely
very small. Very
Yeah.
small. I would like to say I would not recommend this one because it's a very small and just get more like a conceptual idea in the sense. Okay,
Yeah.
when glucose concentration changes, and you can detect by MR somehow it's a glucose chemical exchange sensitive properties using the property.
Yeah. That's okay. Well, that's something also that is something to think about, right? At the limits of our sensitivity, we'll improve our sensitivity, maybe with even higher field strengths or other, who knows? But I think what's useful about that is that it sort of illustrates still that MRI, we're still discovering what, the interplay between physiology and what MRI can see. And this is how it's been for the last 40 years. And it's been still continues on in this sense. And so this is amazing work. And so there's one thing, there's two more papers I want to talk about, then we'll get on to other things. I just have to this is not typical of podcasts where I just kind of list off but you've covered everything, and it's important to actually bring this out. One of the coolest papers I saw in 2021 was the beautiful experiment where you looked at, back to revisit temporal resolution, you looked at the temporal leg, you're looking at cortical or subcortical areas, and you're doing optogenetics to stimulate the cortex to look at that direction flow down to subcortex, but then you're doing sensory stimulation to look at the other direction. And you're able to see temporal differences between these subcortical areas that change direction depending on which end you're simulating from. Did you want to mention that just a little bit? Because it sort of speaks to, it sort of opens up the idea that you can look at directional information flow if you do it right with fMRI.
Yeah. So it's there was my students he was interested to look at the temporal dynamics. So I initially thought about this crazy idea. You've got hemodynamic response. You measure is a dynamic difference there. So he measured anyway. So basically, we do have in our 15.2 mouse fMRI study, we can see activity thalamus and also primary sensory cortical area and the secondary or higher order thalamic nuclei too. So we have a multiple area we see those ones.
Yes.
So when you do the sensory stimulation, we do see somehow is a bold response coming to primary the thalamic cortical area response very first. Then next is a primary sensory cortical area. So you can thalamus to cortex.
Okay.
Then cortex to higher order thalamic nuclei and other cortical area. So you can so initially I thought is okay. One idea you could have it. Well, this is something to do with a hemodynamic response function different area, because we know it's a different area. We have a different response function.
Yes.
And so that's one of the issues. And second thing is we thought about, okay, so we have a primary thalamic nuclei called is a BPL, and the other one is high order thalamic nuclei, actually side by side. They are very close. So should have a very similar hemodynamic response function, but actual fMRI response are quite different. The higher order much lower lag to the primary one. So we thought it's something to do with actually it's information flow.
Yes. Yes.
Well, so then we need a reverse order. So we actually do the optogenetically stimulate the primary motor area. Then this one is project to the primary sensory area. Primary sensory area project to the thalamic nuclei. So reversed order, we actually measure again. Seems like actually our measurement correct. All right. So it's a quite interesting one. So the question is now is how you explain. So we actually do also hypercapnia style. We do the CO2 challenge. We actually do the experiment. So we do all the experiment. Then we also look at the layer. It's the somatosensory area where the layer 4 coming out first. So we look at all of those. So looks like everything match. Okay. Now there is a very fundamental issue is why do we see it? We never expect to see that well in the sense because we are measuring hemodynamic response.
And you're just using bold, actually. Like
we use the bold. We're using the bold. So we're using the bold. And then, so one basic idea is we must see the capillary early time point. So we use a 15.2 Tesla scanner. So it's a very high magnetic field. So maybe like sensitizing the capillary area. So we must detect capillary response at early time point. So that's one is of a conclusion there. Second is why we actually capture this is like information flow order with the bold fMRI. I mean, this is a more fundamental question though. So I don't really have an answer. So only thing I can speculate the idea. When you measure neural activity, time difference, they only look at the first spike. First spike coming to the thalamus, first spike coming from the sensory area. They say it is a 5 millisecond time.
Right. It's so short. Yeah.
It's short. Right?
And these legs are a little bit longer. Right.
These are much longer. So it's basically it's much longer. So now why is it happening? And this is the only thing is you talk about neuromuscular coupling issue. Okay. So it's my speculation is coming out in the mentioned in the paper. Somehow it's hemodynamic response or capillary response is related to the cumulative synchronized neural activity. Okay, if not synchronized, then it's not likely active very easy. So it have to be somehow synchronized. So if you consider that idea, then you coming to the thalamus, to the project to next cortical area. Then there was initially is then is a synchronized signal coming in, then going to the higher order area, a little bit less synchronized. When you go to the next downstream sequence they are less synchronized, probably less activity. Maybe it will take a longer time to respond capillary response. I don't know. I just completely is a speculative argument.
Yeah.
So it's a need like kind of modeling.
Yeah.
I think it's a kind of a neural modeling somehow is okay. Question is what is the induced hemodynamic response? Then you can accumulate those synchrony, all those ones.
So that's interesting. So it seems like one could try to model this like a coupled system with some legs, but then sort of this feedback. And if they then convolved it with a hemodynamic response, can they reproduce your results in that regard? If they tweak like you're saying, like the there's a coupling factor and there's a latency factor that's really short, but the coupling factor maybe might extend out these sort of things.
But I think the synchrony is a very important factor though. So because we actually have there was another faculty in our center there, and she's doing a two-photon study. She looking at calcium activity. When you look at the individual cells in the cell calcium activity, when you synchronize, it responds much faster. It's not synchronized, even though its amount is similar. Then synchrony is low, then you actually respond very slow.
Interesting.
So I think it's somehow cellular synchrony is a kind of important appear to be factors.
Yeah. No, I know what you're saying. I see what you're saying. Huh. Okay. All right. So all right. Okay.
So it's not easy to bring into the MRI measurement because it's not easy. So this is a more like optical, study. And you do the two-photon study, then you look at the KPI dynamics, those one. Then look at all the cellular activity, see how they are modulate in the sense. So you can have same neural activity without the correlation, coherence.
Yeah.
Okay. Or you have, basically you change the coherence somehow.
Yes.
You can modulate the, hemodynamic response.
Yeah, that's hard to get an intuitive sense of because, I'm trying to think of, right. So you're driving the magnitude of the response based on the level of the amount of synchrony, and somehow that's affecting the lag in that regard. It's sort of going.
So that is somehow it's probably synchronized. Then there's a more neurovascular modulators probably released. Right, then it's probably reached a certain threshold to kick into the vascular response.
Got it.
Okay. So if you are not synchronized there, but they release, but they are not accumulating. They probably release it, then it's washed out very quickly somehow. You lose it.
Yeah, I got it. Okay. All right.
So something is like a timing of the synchrony is important. Reason is maybe like a vascular regulator modulator, somehow they release at the same time, then increase the concentration.
Yes.
Okay, then you respond, you hit the vessel dose. I mean, this is completely speculative.
Yeah. Oh, that's okay. So, right. And I think actually that's the thing. I mean, you have this observation that's very clear and it works in both directions. And it sort of forces us to rethink things in this regard in terms of neurovascular coupling and how that could translate to, and I like the idea of somehow, having this accumulation of activity or vasodilator release based on the synchrony sort of generating that, and then that pops up and at just the right amounts of time down the network. I have to think about that more, and I think that's a perfect thing to try to model. It seems like one should be able to just model that and to explain it because it doesn't come easily in some sense as far as the intuition is concerned. But yeah, that's cool. That's cool.
Maybe I think this is what the modeling exercise. So I think it's a neat model. So we have observation.
Yeah.
Then we can match. You gotta change the model somehow to match this experimental data set. Then we can assuming that we can then test out what kind of idea we can try in experimentally. Or like we can test that it's a probably model somehow different way there. Then we can prove it or disprove it model in the sense.
Yes. Yeah. And this is also right. This is what I love about how you go about things is in the sense that it's very right. I mean, every experiment generates sort of some questions, and then you just dig in and you keep on digging as far as that's concerned. There's a couple of things I skipped, but there's so many things you've looked at. One thing that's just want to mention briefly, that's because it's so hot off the presses in terms of you did looking at, you're sort of delving into resting state a little bit. And you did, once again, working with optogenetics, you've done optogenetic silencing of excitatory neurons, and or maybe also, and how did that profoundly have changed the resting state correlations as far as that's concerned? So maybe mention that really quickly. Yeah, PNAS 2022. Yeah, yeah.
Okay. So when you do the resting state, resting state fMRI is very hot now. Like we do look at a lot of correlation, we see the activity in the sense. So its assumption is in the resting state they are fluctuating, and fluctuation relate to the neural communication. That's what is we are underlying assumption. So if you do the silencing one part of the brain area, then that area is whatever network area, then also suppressed together because there was a network in the sense.
Yes.
So we are looking for essentially when silencing part of the brain optogenetically, then we want to see that downstream area somehow is a reduced signal together.
Yeah.
Okay, so I can consider it's a true resting state network.
Yes.
All right.
Yes.
So what is we are doing a typical resting state network? We just look at the correlation. You don't really see the causality in the sense.
Right.
In this case, we knock down one area, we see which area changes. You can actually see the effective connectivity from that stimulation site.
Yes.
Okay, that's what is the whole idea we have with those one. So what we use there, so we can optogenetically, you can silence excited neuron directly or indirectly. So we actually use indirect process. Reason is you excite inhibitory neuron, then inhibitory neuron suppress excited neuron.
Yep.
That is the indirect pathway. And this is what the neuroscientists use very commonly. It's working very well as well. So I think this is how we tease out resting state connectivity. So what we do the experiment, when we do the experiment, so we actually put the silencing primary somatosensory cortex area, primary motor cortical area, and the secondary somatosensory, we silencing those one. Then we actually found the resting state network. Resting state network mostly ipsilateral side of the somatosensory network. That means silencing that only one hemisphere, not the both hemisphere.
Okay.
Including thalamic nuclei, all those sensory motor area. Okay. In typical resting state, you look at the bilateral, okay, and bilateral activity there. So what we actually looking for? Bilateral resting state activity does not really meaning direct communication between two hemispheres.
Okay. Okay.
All right. There is a modulate together, but it's not like a direct, cortical modulation. There is something else going.
Yeah. So if there were this direct connectivity, if you silenced it, it would suppress the other side.
That's right. That's exactly the other side, same amount.
Right. Right.
Yeah. Yeah.
That's interesting. So it's sort of like there's some source.
That's right. Some source going to both hemispheres.
Right. Right. That's great. And I think people have suggested there's some subcortical source and that's the sort of thing, but this is direct proof of that. I really like that. That's a
problem is so far is all the optogenetic stimulation study, they are excite. They excite excited neuron, they look at the network.
Yes.
Okay. So it's our approach is we always interested about the inhibition, because inhibition is useful to dissect the circuit, because you don't inhibit, then you don't know whether single synaptic connection or polysynaptic connection. You have no idea what you're looking for those one. When you're silencing, then you can tease out one at a time.
Yes. Yes. That's really cool. That's very cool. So it seems that it makes me start to think about layers in that regard. You can start to maybe tease apart, aside from, once again, just using resting state and having seed voxels in various areas and see the layer activity, you can start to actually tease apart true functional neuroanatomy in terms of silencing. I don't know how accurate you can get with that optogenetics, but silencing individual layers or and that sort of thing.
You can silence individual layer. They
Okay.
have a transgenic mouse model. They have a certain specific layer, have a certain it's a key line. You can inject the virus in that specific layer, and it's work out. You can just target the specific layers. Oh,
that's so cool. I mean, not only from looking at the connectivity, resting state, and really probing that, but also just looking at the mouse behavior. I mean, obviously they're unfortunately they're anesthetized, so it's hard to do. But
no, you can do the awake study. So what we usually do is we do two ways in the sense. So we do a lot of this study. We do the awake condition is a behavior study. We actually
Okay.
trying to find out what is this is the impact in the whatever you do silencing certain part of the brain, how to change the behavior.
Yes. Yes.
Okay, you know what's going on, then we use the same mouse line. Essentially, we do the fMRI study.
Yeah. Okay.
But it's just like one is awake condition. You know what the property of those silencing in the sense, how is it changing the behavior. Then we gotta combine with the fMRI.
That is so nice. That's there are so many experiments that you could do.
So we have a recently what we do is like we have a chronic pain model in the mouse in the sense. So when we do the ACC, we silencing ACC, then the animal does not feel the pain. Okay, you suppress the pain in the sense. So ACC is a key component, and this is a pain circuit. Then we actually use fMRI because the ACC connects to a lot of different places.
Yep.
Right? Question is what exactly ACC, which circuit is specifically to modulate this pain circuit also.
Yes.
So then we actually then do the fMRI study. Then we find which circuit is likely the source. Then we do another optogenetic study, ACC2, whatever circuit specific optogenetic modulation. We want to see whether behavior keep changing the same manner as ACC modulation. So you can kind of use fMRI used as the kind of tool set to narrow down the circuit in the sense of what the possible circuit for your next research.
That is so cool, right? Yeah, using the general fMRI, narrow it down, do that specific circuit and then look at the behavior, but then you can dig in deeper and look at very specific types of within that circuit. That's cool. That's
what is we actually the labs what the pushing in the direction. So we looking for behavior, like a circuit, circuit determined by, for example, fMRI.
Yeah, that's, and I really don't know. I'm not aware of other groups that are doing that or things. There might be some groups doing things somewhat like it. I mean, people are doing calcium imaging and things like that, but certainly there are groups doing optogenetics, but not in this way. This is really directly relevant to the whole field of resting state and trying to figure out what it means. So this is
I think it's useful to neuroscience community. A lot of neuroscientists don't really care about fMRI research. You talk to system neuroscientists, you talk to them, they say, oh, so we can do these things, why we need to have a fMRI,
Right.
So I think this is what we can offer is we can do the whole brain.
Yep.
It's a network information to them, and that they can get some information what the target is a good target for their next research.
Yeah. So it's a tool for iterating and getting focused on the target. But then also, of course, it iterates back. And then as you know about what's happening, then you look at the fMRI signal and you say, oh, this is a signature of what's going on in this way. So it helps fMRI and fMRI helps this.
That's right. So I think I have another example. I think we did not publish yet there. So we're working on the autism model in the sense. So this autism is a mouse. So they have a hyperactivity. It's a sensory hyperactivity. So we want to know what the sensory hyperactivity means in the fMRI. So we actually did experiment. We actually observed hyperactivity in the sensory circuit.
Okay.
So essentially what the behavior, what they observe in the sense, and then is fMRI measurement. Even we do the anesthetizing measurement, we actually see the hyperactivity in the sensory circuit, and they actually match it quite well.
That's cool. That's great. That's such good work. I mean, because the answers are so clear. I mean, it's like, it's just there. The information's there, and this is what we did, and it has direct implications. So it seems like your work is building momentum. And so obviously, just to mention briefly, to give everyone a perspective, I know we're going a little bit over time, but this is really, so we covered most of the topics, and obviously there's many more we could cover, but that just hopefully gives an overview a little bit about what you're doing. So as far as all the tools that you've used that have been helpful, I mean, you've worked with, everything from 2-photon to optogenetics, optical imaging. So what has been a good complement aside from, so you've been mentioning optogenetics, but other things that have been useful as far as that's concerned?
Well, obviously, optogenetics is very useful. I mentioned already, and I think I found this optical imaging quite useful for us. And even though optical imaging, we can measure calcium, wide field calcium imaging, as well as intrinsic optical imaging.
Yes.
So intrinsic optical imaging is very similar to the fMRI signal, but sensitivity is much better off. So much easier we can try to figure out what's going on before we actually start the fMRI study. fMRI study sensitivity very low. So, I usually actually try to do intrinsic optical imaging first, then nail down what I want to do fMRI study. Then I focus on those subset of the optical imaging study to the fMRI. So I think the intrinsic optical imaging was very useful, and we still actually use intrinsic optical imaging for our research.
Okay. All right. Yeah. And so for intrinsic optical imaging as well, you can potentially be sensitive to calcium.
We actually do the calcium. So we have a multi-wavelength. So we actually have a 3 wavelength system. So we have 2 wavelengths in the intrinsic imaging and 1 wavelength of calcium. So we actually do all this together. So we recently built the optical system inside the MRI. So we have an MRI scanner. Then we built all these things MRI together. So we actually try to acquire whole thing with fMRI together. So it's optics and fMRI, and then optogenetic stimulation. So we have pretty much like what the Yale group did as Lake et al. The papers. So we have a similar system, and then in top of the optogenetic stimulation. That's what we built now, and I think this is going to be presented this upcoming ISMR meetings.
That's great. That's great. So that naturally leads us into, so you went from Minnesota to Pittsburgh, and then from Pittsburgh to where you are right now in Seoul. Why don't you, I visited your center a few years ago, and I was just blown away with everything that you had. It was a beautiful, beautiful facility. Might want to talk about it, maybe just to mention a little bit about what its capabilities are and the people.
Thank you. I think this is a great opportunity to talk about my center, those one. So, I came to Korea like 2013 to set up the new imaging center. When I came to Korea, there was nothing there. So we built a new building. So we designed the building, trying to do neuroimaging research, those one. Then we recruit a lot of neuroscientists. We have a monkey neurophysiologist. We have a human fMRI scientist we recruited there. We also as a computational neuroscientist we recruited there, and we also recruit a bunch of the MR scientists in the center. Those ones. So it's essentially covering about the MR and the optics, and then it's electrophysiology and the cognitive neuroscience research like that. And then we kept the tool set there. So I'm an MR scientist. Obviously, I bought the 4 scanner and the 2 animal scanner. This is my baby in the sense of 9.40 MR scanner and the 15.2 animal scanner for my own research. And I also bought the 2 human scanner, 30 Prisma and the 70 Tera system a few years ago. So we have equipped is a state-of-the-art human fMRI research program. And we also have a 2-photon microscopy system and various optics. And we have a monkey housing facility as well as like rodent housing facility. So we do covering electrophysiology in the mouse and the monkey, and optics in the mouse, and fMRI is mouse, monkey, and the human. So we trying to do more like a cross-species, multimodal, multimodal neuroimaging research in the center. So I think we cover, like more like a technology development, like myself and Camille Uldax. And we have also kind of neuroscientists, they utilize those tool set to do their research, those ones. So I think it's like a combination between, neuroscience research and technology research.
Yeah.
So I got influence a lot from the neuroscientists, because now my research is going to very much have a system neuroscience. Reason is my colleague are the system neuroscientists. They keep complaining about my technology because it does not provide any useful information. So, so I devised the idea of trying to be useful. So I think it's a very good combination between the method development and also it's a neuroscience program.
Yeah,
it's quite exciting.
Yeah, and there's so many tools, and I love how it's laid out with it's not just people doing the physiology and the technical stuff. It's the neuroscientists as well that are really good neuroscientists who are sort of thinking carefully at the limits of the technology as well about what questions they can ask. I mean, it's a wonderful synergy, and it's really nice. And it's a critical mass of people too that you have a lot of buzz of conversations and collaborations going on. So I just think it's just right. I
think it's having everybody in the same place, in the office next to yourself, next to each other. That's very important. It's in the United States, I was there, you are there, and so a lot of good people around there.
Right.
But it's a pretty far away different building, and you need to make a special appointment like that. So much harder to get a casual conversation.
Yes.
So I think what we are saying is all the people who are interested in brain research, whether method or like a monkey or human computation, they are all the offices side by side. I think we do a lot of interaction that make a difference in the sense.
Yeah, that makes a huge difference. And you might be back I don't even know what, as far as Omicron, with COVID and everyone, is everyone back in the office and working? And
yeah. So Korea is never closed also. It's Korea, they control the COVID, handle the COVID very well.
Yeah.
And we never closed there. We keep working. And we actually is working probably better because students cannot, go around. They can work. There's a lot of restriction. What they can do? They can go to the bar. There's a lot of like a restaurant was closed during the COVID time period.
Yeah.
But so laboratory is always open. So
just go to the lab, but nothing else to do.
Go to the lab also. Yeah.
That's great. And you have a great, as far as the layout, it's just awesome too. It's really exciting, and the work you're doing is truly, it's important, it's insightful, it's broad, and it's relevant. It's exciting to me. So are there any, you've talked a little bit about what you're planning on doing. Is there anything else that's like really excites you as far as the potential for certain questions that could be asked, or certain things that are still a mystery, or what excites you most? What do you want to figure out before you retire?
I think a few things that there was. It's a puzzle, but I think it's very important in the neuroimaging community. One is we do a lot of resting state fMRI or default mode network, all of those. What is exactly we are looking for? So I'm very much interested about it's like a resting state is information. What is the biological source of the resting state information? That's one of the things there. The other one is order layer, or we talk about time difference in the sense. Question is what we observe in the empirically also. Look at the very good high resolution, very localized in the input layer. But we know it's neural activity spread all different cortical layer very quickly.
Yes.
As well as we talk about a timing difference. So this thing is, we observe something of an interesting one, but we don't know why.
Yeah.
All right.
Yeah.
So we really do not understand biological source, or it's what is the underlying cause of this is our observation. Observation looks very good for us.
Yeah.
But it's why? I think this is something is unknown. I keep looking for those one, but it's not easy to tackle the issue. Or I don't know how to tackle this issue. So I look at a lot of different ways, optics, everything I can think about, whatever tool set I have experience in the sense, and just not easy to tackle the issue. But resting state, I probably can tackle the issue because we have, silencing method. So we can do a lot of silencing. So we can map it out to the resting state connectivity using the silencing. Then we can change it. So like a lot of different conditions there, we can measure those. So I think that those things I'm still very much interested about the biological source of whatever we are measuring.
Yeah.
okay, on the cellular basis, those ones. So like previously, I mostly look at the biophysical basis, or we look at the product flow, product volume, etc. There. Now I'm more likely what is a cellular or molecular basis of our what is the fMRI signals.
So when you say cellular or molecular basis, you're looking for the specific transporters that relate to neural activity and the whole connection between, neurovascular coupling and with, like you were saying.
Well, so some of the things I cannot do it because this is some more people I'm not expertise about certain area. So it's but if you look at the layer, then optic study is not easy to the layer study.
Yes.
You can. So I think there's a limitation about a certain tool set. So I mean, kind of is still okay. Question is why we see it in the sense of what the recording in the sense neurophysiology study, what people are doing, and what we observe, how we can somehow match or we can explain our observation by the neural activity or neural recording or something else.
Yeah, no, you're right. I think that's everyone's complaint about fMRI in some sense. It's sort of like it's a strong signal. My intuition is that there's a lot still in the signal to pull out, there's a lot of assumptions about specifically what goes into it, and the more you explain aspects of it, the more you open up its potential in that regard. So there's a lot of work to do. You're not going to retire. There's a lot more work. I don't want you to retire. The field can't have you retire.
Well, I don't want to retire. Okay, so I don't want to retire. Only problem is in Korea there was a retirement age there. So, but I don't want to retire because I have still a lot of things to solve it, and I have a lot of ideas of how to do it. So, I have to work. You may hire me. So.
Yeah, we'll hire you definitely. All right, if they force you to retire, we'll hire you, and
I work as a postdoc.
That would be awesome. That would be. Oh God, yeah. So I have to say that your approach is clearly, over the years, it's been inspiring in the sense that you're fearlessly. It's kind of like this fearless sort of like forging ahead, asking these questions. It's very disciplined. It seems like you keep your focus on the interesting problems all the time, as far as that's concerned, and the methods. And so, what advice, just to finish up, what advice would you give to young researchers? I mean, or even perspective of kind of what it took for, of course, we were talking about a little serendipity, but also you've forged this field with your work. And what do you think is essential to be successful in this regard?
Well, I think this is a difficult question. I mean, we all know we have to do hard work, but that's not good enough to be successful in the sense. We all know about this one. So I think it's selection of your idea, a project is very important. Everybody work very hard. We all work hard. All young people or all people, we work hard. So selection of a critical project, something you can solve.
Yes.
I think that it's a very critical. So I already thinking myself, okay, why is it so important what I'm doing? Why I'm competitive, better than other people. Peter can do better than me. I don't want to do it. So that's the kind of idea what I have it also. So I already, whenever I evaluate the project, the question is, what is my contribution to the scientific community? And then whether I can do very well. So you see that my research field is very narrow. You can see it's not deviate because something I know this area I can do very well. So that's why I keep working on these areas, very narrow area. So it's not can be general, but I think it can be anybody. So you can choose a project, something important, and you can do very well. And then you can enjoy what you're doing. I think when sometimes you fail, sometimes you succeed, but during the failure, you can find something new. Because I always thinking, when you try, you fail, you will be getting the very interesting observation. You succeed, you have just papers.
Yes.
All right. You plan to the experiment, you got there and you got just paper, then maybe not very interesting. But something you fail, then you find what's going on, then this is a real interesting.
Yes.
All right.
Yeah.
So that's the kind of keep working. I mean, and then not discouraged about what your failure, and you can try to enjoy what you're doing and try to find something new from your data set. That's what I tell my students. Try to find something new from your data. That is there.
Yeah. So you balance. I mean, I think that what you said is really important, right? As far as you have a new student or postdoc coming in. I love that you mentioned, I mean, certainly it's good to explore, but it's important to have a goal, to a problem, frame it in a problem that you can actually feel you can solve, and then what it means to solve it, and then go about doing that. I love that focus. And I think a lot of people actually don't necessarily go in with that sharp sort of focus in terms of the problem with the tools to solve. And then, of course, like you said, you learn a lot from your data, even if it isn't successful. Yeah.
So you see that like a lot of people thinking is, okay, I like to do what I like to do. So this I'm interested in this one, I want to do it. So I said no, because you are interested, that does not mean you can succeed this one, because that's not out of your expertise or out of your research area or out of your ability. Because I want to be a singer, even if I like to do singing. Does not mean I can be the singers. Even if I practice a lot, does not mean I will be singers. Yeah. So I think the same thing as every research area. So if you are very good about certain area, then you focus on those area where you are doing the modeling very well. Then you focus on modeling. Yep. You do the MR per sequencing is very good. You can focus on sequencing very good, processing very good, and I think you can utilize your expertise in the sense, or what you are good about those.
Yeah. So playing to your strengths as far as that's concerned, and then it will sort of expand out as you delve into it. But yeah, starting out with kind of what you can actually hit the ground running with in terms of your strength. Yeah. Mm-hmm. Yeah. Well, great, inspiring work, and thank you so much for coming on. We're obviously spent more time than I expected, but it's been amazing, and it's sort of. It feels like it just passed in 5 minutes, but yeah. So this is great. Well, thank you very much, Seong-Gi. I really appreciate it. And
thank you so much, Peter. Thank you.
Best of luck. Yeah. All right.
Neurosalience is brought to you by Organization for Human Brain Mapping. This week's episode is produced by Ömer Faruk Gülban and Stefania Assimopoulos.